Internal structure of leaf in cycas. As we have discussed that cycas exhibit dimorphic leaves, which are foliage or asymmetry leaves and scale leaves. Foliage and asymmetry leaf has an pigmentally compound arrangement where leaflets are arranged over the edges on the adaxial side in an opposite arrangement. Foliage leaf has two parts, a rachis, a central axis over which leaflets are present. The internal structure of rachis. It is circular in outline and has two rows of leaflets present on the adaxial side. It is differentiated into epidermis, hypodermis, ground tissue, and vascular tissue. Epidermis is the outermost layer and is covered with thick cuticle. It is interrupted by sunken stomata, which consist of two guard cells and two subsidiary cells over the lower side. Hypodermis is made up of two types of cells, as you can see in the diagram, chlorenchymatous cell and followed by splenonchymatous hypodermal cells. Ground tissue is composed of parenchymatous cells and it is transversed by many mucilaginous canal or ducts. There are vascular bundles present in the ground tissue. Here the vascular bundles are arranged in an omega-shaped arc-like structure, and this arrangement is a very characteristic feature present in cycas. So, vascular bundles are conjoint, collateral and open, and each vascular bundle is surrounded by a single bundle sheath. There are diploxylic vascular bundle where centripetal xylem and centrifugal xylem here coexist with each other. You can see in the diagram, centripetal and centrifugal xylem are present and they are present within the phloem in the inner side, so it forms a conjoint and collateral arrangement with a cambium present between xylem and phloem called as open vascular bundle. Now, as we go from base to apex in the rachis, suppose in this diagram as we go from base to apex, so there are difference in arrangement of xylem and phloem as we move forward toward the apex. So, the basal region, the vascular bundles have only centrifugal xylem and they are endark in nature. There is no centripetal xylem here. If this is the phloem and this is the xylem, then there is only presence of centrifugal xylem in the basal region. And the xylem is in endark condition where protoxylem is in center and metaxylem is towards the phloem towards the outer side. Coming to the middle region, slowly protoxylem cells, some of the protoxylem cells move forward to the lateral sides, lateral direction, and finally change into centripetal xylem. So in middle region, both centripetal and centrifugal xylem are present and phloem is present on the outer side. Whereas in upper region, centrifugal xylem get reduced and is present only in small groups or patches, and centripetal xylem is present more. And therefore, at this stage, there is more centripetal xylem than centrifugal xylem. This is the diagram of internal structure of rachis. We can see the omega sheath structure formed by vascular bundles, which is a characteristic feature of the cycas rachis. Internal structure of leaflet. Internal structure of leaflet, if we talk about the transverse section, so we can see the upper and lower epidermis, where in leaflet there is only single midrib present and there are no leaflet veins. So here this is the region where midrib is present and the lateral region is called as wings of the leaflet. The epidermis is the outermost layer and it is the upper epidermis is continuous, whereas lower epidermis is interrupted by the is stomata, which are present on the wings of the leaflet. We can see here the interruption and there are the stomata. The hypodermis is composed of sclerenchymatous cell, and over the dorsal surface there is a continuous layer, but on the ventral surface they are only confined to the midrib region only. Now, they function to protect the plant from the excessive heat and transpirational activity. Next to the hypodermis, you can see it is followed by a mesophyll tissue which is differentiated into palisade, which is a columnar, regular arranged, tight arranged cells, which are then followed by spongy parenchyma, which are loosely arranged oval-like shaped cells. So mesophyll is differentiated into palisade parenchyma and spongy parenchyma. Spongy parenchyma are confined to wings and are rich in chloroplast. Now, after mesophyll, we can also see a characteristic feature called transfusion tissue, which helps in the lateral conduction of water and nutrients in the leaflets. So, the transfusion tissue are nothing but trachea-like cells which are present on the lateral side of the vascular xylem, and these are colorless and transversely elongated, which help in the lateral conduction of water and nutrient as they are connected with the centripetal xylem. In the diagram, upper epidermis is covered with cuticle, followed by a hypodermal layer, followed by a mesophyll tissue differentiated into palisade and spongy parenchyma, a transfusion tissue which is confined to the midrib, present at the lateral side of vascular xylem and is connected to the centripetal xylem. Then it is followed by the lower epidermis interrupted by the stomata. Now, vascular bundle in the leaflets they are present in the midrib region. They are surrounded by bundle sheath. They are conjoint, collateral and open, means xylem and phloem are present on the same radii, and the xylem is present towards the inner side of the phloem, and diplozytic means that centripetal and centrifugal xylem coexist with each other. Centripetal xylem are larger and triangular, and two small groups of centrifugal protoxylem are present in the vascular xylem system. So, one on these two centrifugal xylem, one on the either side of centripetal protoxylem are present. Phloem is present on the adaxial side of the xylem, and this forms the complete vascular system in the leaflet. Diploxylic vascular bundles are present with both centripetal and centrifugal xylem. Thank you.